Hello, in this video we will discuss about the tissue culture of plant, and we will also understand about the difference between the cloning of the tissue culture and recombinant tissue culture. So let's begin to understand of the step of tissue culture, and we will also understand about the preparation. So the step is the preparation media composition. Media composition mean the composition of the nutrients which that is equal to the natural nutrients are present in the soil. For example, the vitamin as well as the organic substances. So another is the basically uh, dealing with the contamination. So the contamination should be free, and is the next. Here we will discuss about the preparation first. So the medium should be same as natural environment of wild, but also we are checking that media composition. So the media composition is basically is not completely equal to the soil or the wild, but it is close to equal. So here is the plant species. When we can get the desired plant species, and we will separate the tissue from the parenchyma, which that is the living cells, and we will separation of the tissue. After the separation of the tissue, basically agar gelling agent we will prepare, and that is in the form of powder, which that is mixed with the water to show the gel-like structure. On the other hand, each species want unique media. However, the general stock solution consists of organic elements, micronutrients, and macronutrients. An aseptic technique should also be used for all culture medium preparation. So that means the micronutrient and macronutrients mean the glucose and amino acid, etc., is the macronutrients, while the micronutrients basically the minor quantity in as compared to the macronutrients. So the micronutrients can be metal ions as well as salt and mineral. So on the other hand, the proper preparation of media we will discuss here. So that means the mixed powder media with appropriate of water 800 ml, which that is we use for 1 liter medium. So it means the 800 ml water will be used. Fill a beaker 800 ml water, and 30 gram sucrose we will use in the third step, and set the pH at 5.8. 5.8 means the less acidic but near to the acidic basically. So add add agar to the beaker 8 gram. Add hormone if required the hormone, for example the rapid growth by a oxygen or something. So add 200 ml water also, and autoclave this media, and dispense the melting media into a sterile tube, and make sure that each tube is each tube is labeled, and we can identify the each tube which that is contain a medium, and we will put the tissue, for example one cell protoplastic cell, which that is basically living cell, is used for the tissue culturing. So the last step for the preparation of media, the media should be left in a sterile environment. So it means the contamination free should be. On the other hand, here is the three medium containing test tube, which that is used for the, for example I am giving the example. So it is used for the tissue culturing. So the plant tissue culture media are generally sterilized by autoclaving at 121 degrees Celsius, that is a huge, and 1.05 kilogram per centimeter square, which that is used equal to the basically 15 to 20 psi. So the time required for sterilization depends upon the volume of medium in the vessel. So the autoclaving is used for the sterilization process. So on the other hand, when we will take the test tube, basically contain a nutrient media for tissue culturing. We will separate the different species containing tissue, and we will when separate that different tissue containing species containing tissue separation, so we will put the either multiple cells or one cell for growth, so that sterilization of X plant contamination should be free. And on the other hand, the nutrients media used for the growth on the basis of normal pH is the basically is, range is to 5.8, as well as the temperature is also important for it. So the temperature should be low as compared to the environment, because the tissue culturing requires the sensitive temperature and sensitive nutrients for the growth of the of the tissue to become an embryo. So we are putting the nutrients media with containing tissue culturing tube set in growth chamber. So when we will put the set into the growth chamber, we can put the sunlight near to the sunlight, or on the other hand we can use the electric bulb for use, and carbon dioxide moderation and clean environment should be. Why? Because during the early growth of the plant is should be contamination free. Why? Because it should more sensitive of the pathogen, so pathogen can infect. So that means pathogen free should be environment we can provide. And on the other hand, here is the plantlets are regenerated from X plant, so we can generate after a week the development of tiny plant will be occur. So this tiny plant is basically a clone of our desired traits. This plant will same genetically than the parent plants. So it means the genetically same. Why? Because the tissue culture containing a same nucleus. So it means the cell division will remain constant and the mitosis will keep this process. So this is known as the asexual reproduction. It means the haploid cell or gamete is not formed. So it means the diploid cell is again grow. So that is why it is known as the basically as asexual reproduction, which that is part in our desired. So that is the cloning mechanism. It means the clone we can produce. We can produce clone of the parent plant. Why? Because we want to desired character. So that is why if we want to desired character, so we will choose the tissue from the desired plant or X plant, and that desired plant is important for us and we can grow again in the laboratory. So that is all thing is laboratory. After the plantlets growth, after the plantlets will be grow, we will put the into the greenhouse. Why? Because it remain the carbon dioxide is high, heat traps don't allow to sunlight escape. So it means the outer environment if the less amount of temperature, so it means the inside the quantity of the heat is more. So that means the plant will grow easily and plant will enjoy to grow. So the sunlight escape is basically sunlight will not escape. Why? Because greenhouse, which that is sheet is allowed to inhibit the sunlight radiation. So that is why. That is important things to understand. So that was the cloning, but we will again understand the gene of interest in tissue culturing and transgenic plants. So we will understand the gene of interest in tissue culturing. So we can put the gene into the into the normal plant to become a transgenic plant. So how it will be produced? Let's begin to understand. So here is the plant. This plant is resistant to pests, herbs and chemicals, for example. So we want it for another non-resistant plant. So that means we can apply another species these genes. So we will separate the cell tissues. So these tissue contain a cells. When we will separate the one cell, this cell is looking like this, and this contain a messenger RNA in the cytoplasm. Why? Because it is gene active in the messenger in the form of messenger RNA due to the transcription, and that means we can say that the gene is active. So that is the basically pest, herb and chemical for example that is resistant for that gene. So when we will separate the messenger RNA, that is known as a pest resistant gene containing messenger RNA. So when we will selective primer which that is prepare in our laboratory, which that is a complementary of the messenger RNA for our desired. So then when we will produce the primer by this process in the laboratory. So this primer will begin by the polymerase enzyme to begin the reverse transcription. So reverse transcriptase enzyme will be used. So the complementary DNA will be formed. This complementary DNA is the basically is the contain a uracil. Why? Because the RNA. So we can use the RNAs to digest the uracil and again produced by the polymerase enzyme to complementary DNA by attachment of the primer. So these complementary DNA used into the thermocycle for amplification of the desired segment is known as a PCR, polymerase chain reaction, will produce the many copies of the gene, which that is known as the amplification of DNA, which that is contained three steps, basically denaturation, annealing and extension. So here is the another plant. This plant is non-resistant or non-resistive. We want to improve this species and plant name is we are putting the lambda. So basically we will separate this tissue and this is basically non-resistive. So that means we will separate the cell wall because it is dead, and we will remain the another part is known as the protoplast. This protoplast is ability to grow again. So remember about that. So when we will put that gene segment which that is our desired, we will put that gene into the solution, and we will also put the protoplast in it. So after the putting of the protoplast into the medium, we will give the electric current, which that is voltage, give the electroporation. So that process is known as electroporation, or we can use as a gene gun or another mechanism to insert the DNA segment, which that is a desired DNA, which that is we are amplifying that gene. So holes will be created. So this hole is looking like that, and the DNA segment will be moved inside, and the ligase enzyme is also used for it. So the nucleus will contain again the recombination of DNA in the whole genome of the cell. So after this, we will improve. We will say that it is recombinant cell. After this recombinant cell is protoplast. When we will give into the tissue culture, it will be grow again the cell wall. Why? Because the gene is active in the protoplast to produce the cell wall, and it is produce the embryo formation. After the embryo formation, that is again the lambda species, transgenic recombinant plant of lambda species will be occur. So this lambda species again, it is same like a lambda species which that is not resistive. So it is resistive, and we can use for the advantages of the resistant genes, productive in productive and increase desired color pattern and quality are many things which that we can improve it. So anyhow, I hope you make sense about that. So the important thing is that the transgenic plant uses agribusiness company process of developing uh, transgenic version of wheat and rice in addition to corn, and some corn and corn cotton plant are both pest and herbicide resistant. An improvement still contain for shield of amino acids, starch, fat, oil, food, vitamin for health issue, and basically used for the mankind. And that is important thing is I am saying about that. So what what is about the tissue culturing of plant, cloning and recombinant. Tissue culturing is basically is the tissue again the cloning and copy of the gene will be produced, and again the desired plant will be again produced by the tissue culturing due to the excision of the tissue. So, on the other hand, the gene of interest in tissue culturing transgenic plant is another way, which that is we can introduce the gene, foreign gene into the plant for growth in the basic function of the tissue culture. But on the other hand, we can also use the plant for plant whole plant to transgenic plant. So thanks for watching.